that's what they're doing. Maybe not.
That's it.
Hello and welcome to CSI of the Sea here at the Zoological Society of London. My name is Charlotte and I will be your host for tonight's event. Tonight we're giving you the opportunity to see behind the scenes here at ZSL and watch a porpoise post-mortem taking place. For those of you who aren't aware, ZSL, the Zoological Society of London, is a learned society and charity, first established in 1826. We run 2 zoos, one here in London and one at Whipsnade. And we are also involved in conservation programs in over 50 different countries around the world, as well as having over 100 researchers based within our Institute of Zoology. And it's these scientists that you're going to be meeting tonight as we find out more about this porpoise post-mortem. We do encourage your questions during this evening's event, so there will be moments when we pause to take them, so please do feel free to pop your hand up if you're joining us here at the zoo, and we'll come over to you with a microphone so we can hear your question. And if you're joining us online, then do feel free to send your questions to us using the email address learning@zsl.org. And feel free to share your comments as well on Twitter using the hashtag CSI of the Sea. So this evening, we're joined by 3 scientists, one of whom is with me now, Paul Jepson, who is a reader and a vet based within the Institute of Zoology, our research arm here at ZSL. So thank you very much for being with us, Paul. And you're part of the Cetacean Strandings investigation program, which is the team who are involved in these post-mortems that take place of cetaceans. So I guess, Paul, the best place to start is to explain what exactly do we mean by the term cetacean? What kind of animals are these?
Okay, so quite simply, the cetaceans are the whales, the dolphins, and the porpoises. So that's what we'll be talking about here, and particularly the porpoises tonight, which are the smaller of the cetacean species.
And we're going to be trying to focus a little bit, in particular, on pollutants and the impact that pollutants have on these animals this evening. So when we're talking about pollutants, Paul, what do we mean? Are we talking about the rubbish and plastic that we see on the beach?
Well, pollution is quite a broad term, but really what we're talking about in this context is chemical pollutants, things that we can get inside the body and have a harmful effect. And the harmful effect really depends on how much of these harmful substances are able to get into the body, usually through the food chain. So that's where we are, and we study a whole range of different types of chemical pollutants in our work, and we'll talk about some of those tonight.
Absolutely. Well, let's now, hopefully, fingers crossed, go over to our live link to the post-mortem room here at the Zoological Society of London, because it's there that we're being joined by 2 of our other scientists, Rob DeVille and Matt Perkins. Now, it might take a moment for it to come up on the screen, so we'll let the sort of tension build. But hopefully, there we go. By the magic of technology, we're joined by Rob on the right there, and also by Matt on the left, and this evening's porpoise as well. So we'll find out a bit more about that in a moment. But Rob, you're the project manager of the Cetacean Strandings investigation program. You spend a lot of your time sort of chasing across the country, picking up these animals that have stranded around our coastline, and you regularly carry out post-mortems or necropsies, as we call them, with animals here at the zoo to find out and try and find a reason for why they stranded and died. Can you just sort of tell us briefly, what is the Cetacean Strandings program and why was it set up?
Okay, hi Charlotte, hi everyone. So the CSIP, and basically it's an excuse to have CSI on your top, so I have to put that out there straight front, first of all. It was set up in 1990 by the Department of Environment to monitor strandings around the UK coast. And essentially it's a consortium of partners, ZSL, NHM, SRUC in Scotland, MEM in Wales, and we monitor strandings, we record them around the UK coast. We pick up a portion to investigate at post-mortem, like this animal in front of me here tonight.
I've got some images, Rob, so we're just going to continue to listen to you, but not see your face just for a moment. Because
probably just as well.
We've got those groups that obviously support your work and all work together very much across the country to try and find out more about these animals. But also you were mentioning what that work actually involves and going out and picking up these animals as well, Rob. And we're talking about stranded animals. So are these just ones that wash up on the beach?
Yeah, so I guess it might help to put some context on this. So every year around the UK coast, we get around 600 strandings of cetaceans, which Paul's already defined. And we're contracted to retrieve about 100 each year to carry out post-mortem examinations on those. And I guess of those 600, around 10% might be live strandings. So actually the vast majority are animals that wash up dead. And I just touched briefly on the setup and structure of the project. To confess that out a bit further, it's funded by DEFRA at the moment and the devolved governments of Scotland and Wales. There are 4 main partners involved in the project, but beyond that, there's a whole range of statutory agencies, voluntary groups that all get involved in stranding response. From everyone, like a guy walking a dog on the beach, all the way up to agencies like JNCC that manage the contracts on behalf of DEFRA. So there's a big involved network of groups and individuals that get involved in stranding response around the UK.
Rob, we've got an image up at the moment of you and Matt actually on the beach with a far larger specimen we're going to be looking at this evening, carrying out a necropsy, a post-mortem actually in situ. What can these post-mortems tell us that we wouldn't necessarily be able to find out otherwise?
Perhaps it's worth making an initial point that strandings have always happened, and they will always carry on happening. Where groups like ours come in is to try and investigate those strandings to work out why the animals died, whether there's a man-made driver to those strandings, if you like. So we're looking at man-made drivers of mortality, things like bycatch, where things are accidentally caught in fishing gear, ship strike, impacts of marine noise, and obviously impacts of marine pollution, like we're discussing. So as well as the cause of death, that's the main reason DEFRA fund the research project. We can do much more than that. These animals are incredibly hard to study in the wild. They spend a fraction of the time at the surface. So although it's a very sad event when you get a stranded animal on the beach, it gives us, the stranding scientists, a unique chance to try and learn more about species which are hard to study in the wild. So during the course of the post-mortem examination tonight, we're collecting a whole range of samples that will help inform research here in the UK and internationally, so we can learn about their diets, about their life history, about their population structure, a whole range of things that are only really informed through strandings research.
Rob, we're getting a few technical issues here, just in that your sound no longer seems to be quite correlated with the movement of your mouth. So what we might do is try and bring you down for a moment to see if there's anything we can do while you're offline, as it were. And this is partly why we have Paul here, so that he can step into the breach if necessary. Because, Paul, the next question I was going to ask Rob was just a little bit more about porpoises, and harbour porpoises in particular, which is the species of cetacean that we're looking at this evening. Are harbour porpoises quite common around the UK coast?
Yes, they are, Charlotte. The harbour porpoise is the most commonly stranded species that we have in the UK. So about half, slightly more than half of all the strandings that we get are harbour porpoises, and probably about a half of all the necropsies, the post-mortem examinations that we do, are also of harbour porpoises. As you can see from the map here, we get the red dots are from one of our reports from the period 1990 to 2014, if those can't see at the back. So in that 14-15 year period there, you can see red dots all around the UK, from Scotland and Shetland and Orkney all the way down to Cornwall, England. And that's very good for a lot of reasons, because porpoises tell us about the health of the ocean. You know, these are animals that are all around the UK. If our oceans are healthy, then our harbour porpoises, we would hope, would be healthy. If we've got any particular problems in any of these regions with their particular threats, we've mentioned some already, like chemical pollution and fisheries bycatch. Well, we can actually see if some of these regions have more causes of mortality than another, and then that's the sort of thing that actually DEFRA and the government want to know. They want to know, okay, what are the causes of death? What are the threats? And are any of these threats more prevalent in a particular area than any other? We're also talking about chemical pollutants tonight, and the good thing about seeing this map now is that these red dots are everywhere, and if you're actually monitoring chemical pollutants, and particularly trends over time, we want to know not just what the levels of pollutants are in any individual animals, any of these individual red dots, but we also want to know throughout the period from 1990, when the project started, all the way to the present day and beyond are the trends. And what we really want to see is some of these pollutants coming down in levels, in concentrations. So a lot of these pollutants have been banned, and the levels are coming down, but not all of them. And that's one of the things that by having harbour porpoises that strand all around the UK, we can see, okay, are some levels more polluted than others? And there are, we can talk about that a bit later. So actually it gives us this very long coverage, a very large sample size, and that's very good for science. So lots of red dots is good. That means that we get a lot of data from harbour porpoises, we know a lot about them, and actually they can then inform the people who fund our research in terms of what, as I say, what the threats are, but also things like what the trends are in things like chemical pollutants and other things as well. Are our oceans getting healthier? And that's what ultimately we want to see.
So this gives you lots of data, plenty of information to try and then inform our understanding of what's happening out there. And I guess too, it's worth pointing out that all these dots, I mean, it could look like we're having like mass porpoise mortality, but this is because there's a large harbour porpoise population around the UK, so it's sort of a sign that there are lots of these animals out there.
That's true. There's a correlation, a crude correlation perhaps, but a correlation between the stranding rates and the population size. So if we have a larger population, then you get more strandings, and that's normal. And actually for some species, not necessarily the harbour porpoise, but some of the large whale species, we have actually seen increases. Like a humpback whales is a very good example. When I first started, for the first 10 years on the project, I started in about 1993, and for the first 10 years I never saw a single humpback whale stranding. But now we get humpback strandings regularly. And so for the individual animals, that's very sad when we do get them. But what it tells us, we know from the population in the Atlantic that we're not hunting them anymore. The whaling industry has left humpbacks alone for a long time now in the Atlantic. And actually the population is recovering. In fact, I think in the US quite recently, in the United States, they've actually, or they're about to take they're thinking of taking the humpback whale off the Endangered Species Act because the population in the Atlantic has recovered so well. It's not the case for some of the other species, but we do hope that over time, all these species that are no longer subjected to very intense whaling operations will recover, but that does mean that we will get more strandings of them, but that isn't necessarily bad news. It can be good news. And in fact, actually, some of the species that strand very rarely, that we used to have probably very common in our waters, those are the ones that I'm more worried about. So I like to see more strandings of humpbacks. I think it's a good news story in the main.
Yeah. Well, I'm just going to glance up. Oh, I'm getting lots of thumbs up from the back there. So we're going to try and go back over to Rob and Matt and see what they're looking and sounding like now. So let's give it a few moments to change over.
I hope they can hear this.
There we go. we can hear you now. I wonder if there's a sound delay, though. Hello, Rob. Can you hear us?
Paul's going to be feeling like that now.
Oh, dear. Okay. We might need to perhaps bring them back down for a moment, I think. And what we can do is we can jump ahead a bit, talk about the pollutants that we've been referring to this evening, and see if we can get them back in a bit. So, Paul, we are focusing in particular on pollutants in our seas this evening. We could obviously talk about all sorts of different threats to the animals that live in our oceans, but we're focusing on pollutants. And you mentioned before that these are things that can potentially make animals unwell and can even be fatal as well in high concentrations. And one of the pollutants that's often referred to now in the press and media when we're talking about strandings are PCBs. What are PCBs?
Okay, PCBs are polychlorinated biphenyls. That's what the PCB stands for. It's a big, long name. So let's forget about the big, long, horrible name, and let's just call them PCBs. Everyone refers to them as PCBs. But actually, these are types of organochlorine pollutants, similar to some of the organochlorine pesticides like DDT that you will have heard of. And they've been banned for many years. They were used in industry. They weren't not really in agriculture. They were used in industry, in electrical transformers, in manufacturing, and also even in tower blocks. So you find them in lots of places on the land, but unfortunately, because they were designed to be very persistent and stable, very chemically stable, that actually they're able to actually survive an awful long time. And even though they've been banned many, many years ago, decades ago really, they're actually still occurring at quite high levels, particularly in the marine environment where they're able to actually accumulate up through the food chain because they dissolve in fat. So that's the type of property that you get. You can see here we get our pollutant sources that mainly come on land because, as I say, these are industrial compounds, industrial chemicals really. And then you can see that this is some of the pathways of them getting into the land. And one of the ways that they get into land is actually from sediments in rivers and estuaries, where they then may settle on the bottom, but with movement of water and with currents and also with dredging sometimes you can actually stir up the sediments, and that means that these PCBs that would otherwise be sort of buried in the mud, they're then mobilized and can go up through dissolving the little fish and the crustaceans, and then at the top of the food chain you get dolphins and killer whales and to some degree our harbour porpoises as well. And so, and because they at different levels in that food chain, you'll get the very highest concentrations at the top. So the killer whales are the most PCB contaminated species on Earth and have been for many years and continue to be so. We're really worried about those, but we're also seeing effects in our harbour porpoises, and that's what obviously we'll focus on this evening.
So it can build up, basically, up the food chains. As you said, these larger animals are then the ones that really suffer because they have such high levels inside their bodies. What impact does that have on these animals then? Sort of what evidence do you see of this?
Well, we know this from a lot of sort of work that's been done over many years in toxicology from the 1960s onwards, that PCBs can suppress the immune system, so it makes animals, if you have a high PCB concentration, it can make you susceptible to infectious diseases. So this is what we can see now is this is a stomach of a porpoise that had a very high parasite burden in. This is Anasarcus simplex is the name of the worms there. It's a horrible picture, a bit like a bowl of spaghetti, but it's inside your stomach. But this is the sort of thing that PCBs can be exposed to. These parasites are in the wild. These animals are exposed to them. You can even get human cases with this, with anasarcus, because it's widely, it occurs in many species, not just harbour porpoises. But this is the sort of thing that if your immune system is not absolutely tip-top, then you can actually succumb to quite higher parasite burdens. And in fact, one of the most common causes of death we see in our harbour porpoises is infectious diseases. And in that group, we also see often higher levels of PCBs, much higher levels than the healthy animals that die of other traumatic causes like the bycatches and some of those. So we think that the evidence is consistent with an effect of suppressing the immune system. But there are other effects that we're probably even more worried about than immunosuppression.
And are we likely to see parasites like this in this evening's porpoise?
Yeah, well, I mean, who knows really? I mean, hopefully we'll get the video feedback very soon. And then the thing is, we don't know what this animal has died from. You know, it's as much as a surprise to you guys actually as it is to us. So we'll see. But we might see some parasites in the stomach. We might see some parasites in the lungs. Harbour porpoises often can have about 4 different species of parasites just in the lungs alone. If they're healthy animals, they kind of tolerate them and they don't get too high, their concentration. But if they're a very diseased animal, or perhaps if they're immunosuppressed by chemical pollutants or other factors, then you can see these very high burdens. So we don't know until we open up. But this is the sort of thing that we would see.
You've got an idea of what to look out for now, because I think we're going to bring Matt and Rob back again. And can you both hear us?
I can hear you.
Hey,
can you see us?
That's sounding
Hello.
good. We can hear and
Hello, Houston.
see you.
Hear me now?
So we've just
Right.
been having a chat about pollutants and the effects of PCBs. But Rob, you are standing right by the porpoise that we're going to be looking at this evening. Can you tell us a little bit more about it and its history? Where was it found?
Sure. So this porpoise lies stranded and died on the beach on the 3rd of August, I think it was, at Carnarvon in Gwynedd in Wales last year. And it's been held in frozen storage since then at our colleagues' facility in Wales, and then more recently over here at the Institute of Zoology. It's been defrosted just in time for tonight's event.
Fantastic. I'm just showing a map of the location it was found, Rob, and also the beach that it was found on as well. You've obviously had an opportunity now to take a closer look at it. What can you tell us about it so far, just from examining it externally?
Okay, so before we started tonight's event, Matt and I did an external assessment. We've taken a range of photographs. We've done some standard morphometric measurements. We do a length, a girth, and a weight. And having done the external assessment, there's nothing visibly at issue with this animal beyond the fact that actually it's in moderate nutritional conditions. Depending which camera we're on, maybe you can see there's some necking just behind the head, and there's also some flattening of the dorsal flanks. So cetaceans should be nice and streamlined, hydrodynamic to go through the water column quite well. And so rounded would be good nutritional condition, flattened would be moderate, and then hollow would be poor. So that's a very quick qualitative assessment of the nutritional condition of the animal. So it appears to be nutritionally compromised to some extent, and there may be an underlying reason for that, which we'll learn more about, hopefully, as we go through the post-mortem. I mean, I should just also point out, which I would have done at the beginning, but we lost the video feed. So Matt and I are ologists, so Paul back in the auditorium, he's the vet, and only Paul is legally able to give the cause of death of cetaceans that we examine at post-mortem. So Matt and I are carrying out the dissection. Together with the data we'll get from the bacteriologist next door, that will give us an overall picture, hopefully, of what happened to this animal.
We should also just point out as well, I'm sure you're all prepared for a little bit of blood, which we'll be seeing in a moment, but because this is an adult female porpoise, Rob, does that mean there's a potential that she might be pregnant?
Possibly. I mean, given the time of year, it's perhaps unlikely. So I didn't hear a lot of the audio because that cut out earlier. Paul might have been talking about porpoise distribution and their biology and distribution and so on, but they tend to calve May to June. So they found it in August, so it's perhaps unlikely it'd be pregnant. There's certainly a possibility it may have aborted as a fetus recently, but we'll know more about that as we go through it. And perhaps it's also just worthwhile just show you how we sex cetaceans. So to roll it over onto this side, we can see in this individual there's an anogenital slit, two smaller mammary slits either side. That's obviously how you make sure it's a female. And with males, you get an anus and a genital slit further forward. All cetaceans are the same from a whale, largely, to porpoises, and that's how we sex cetaceans.
Brilliant. Well, now you've done your external examination, Rob, what happens next? What's the next step in the process?
Okay, so we always examine animals in right lateral recumbency, so lying down its right side. Just like us, their hearts displaced laterally towards the left. So we roll it over, and then we'll begin, if we're at that stage now, we have lost some time, by taking a strip of blubber off the outside of the body. And we'll then measure that at 3 points. We'll measure it dorsally, laterally, and ventrally. And this will give us some numbers to go into our database to work out perhaps how nutritionally compromised it may or may not be. Okay, so Matt will make a note of this. We've got 9, Matt, yep. And 8, and 8 again. So the blubber layer is interesting. You can see, hopefully the camera will show you that the skin is actually quite thick, and the blubber layer is an extension of the skin, essentially. And it serves two purposes. It's insulation, to keep the animal warm when the water's colder, and it's also a nutritional store, energy store that can draw down on in times of nutritional stress. So this stranded in August, and even for the UK, the water should be a little bit warmer, so that's when the blubber layer is at its thinnest. That being said, this is still quite a thin blubber layer, so it has lost some blubber condition. Okay, we'll discard that sample. And next, we'll just begin by taking a range of samples. We'll start by taking some skin, which is taken anywhere in the body. And this can be, will be archived in our extensive tissue archive here at ZSL. But this is essentially used for research to look at how this animal fits in the overall population structure. We're doing genetic analysis. So we will see how this porpoise varies and compares with other porpoises examined over the last 25 years. And then next, just behind the dorsal fin, we'll take probably one of the most important samples we'll collect, which are 2 blubber samples, 2 full-thickness blubber samples. And this is probably very relevant to tonight's discussion because these are samples where we'll analyse the man-made contaminants or pollutants. So I don't know, Paul, if you've said something about this already, because obviously we lost the audio feed for a while.
Yeah, we've already spoken about PCBs and how they can build up inside the body, especially of larger predators as well. I will just point out, I noticed that Matt, who's a senior technician here at ZSL, is wrapping everything in metal. Is there a particular reason for that, Matt, if you can hear us?
Yeah, so for toxicology samples, we wrap them
Oh,
in
hang on,
foil
Matt.
because
Wait one
plastic
second, you're just
bags
coming
would
up
be
quite
stored in a very
quiet.
similar
There we go.
compounds. Did you hear that?
Right, we can hear you, so yeah,
Okay.
tell us about it.
Yeah, so the samples we wrap in tin foil will be for toxicology. It's because the bags that we place them in are very similar to the compounds we're testing for, so the foil isolates them to a degree from this and doesn't contaminate our samples any further.
Okay. And Matt,
Yep.
Rob mentioned how a lot of these samples too go into our archives here at
Yep.
ZSL, so they can be accessed by scientists both here, but presumably also anywhere over the world. Do you have any idea how many specimens we currently have in our collections?
We've got over 100,000 on site, to be honest, a mixture of frozen and formalin fixed, so it's a lot of samples to look after, to be honest.
Yeah.
No pressure there at all.
I bet, no. Fantastic. So, Rob, coming back to you, you seem to be doing a pretty quick job there. You make it look very easy.
Don't hang around. Yeah, we've got to get on with it. We've lost some time. Yeah, so what I'm doing now is what's called flensing. So I'm removing the blubber layer from the left side of the body. That'll expose the body wall, and then we'll take some other samples. We'll take the ribs away, and then we'll look actually at the organs internally and examine them one by one. So from my perspective as a simple ologist, pathology is a description of abnormality. So in what way are things outside of the norm? So what I'm removing here now, having taken the blubber layer away, is the scapula. And that's essentially your shoulder blade. So for those at home and those in the auditorium, if you do that, you can feel the movement of your arm within the joint. That's a ball and socket joint, fits into your scapula as well. Very similar in cetaceans. If I remove that scapula and point it towards this camera, hopefully you can see there's this articulation of the pectoral fin. This is the ball and the socket joint. So a lot of commonalities between cetaceans and ourselves as well, to some extent. Okay, and then we've taken the blubber layer away, and exposed the underlying tissue. What might be evident now to the audience is that the muscle is actually very, very dark. So there are two main muscle groups, if you like, in cetaceans. The longissimus dorsi, which runs on the back from the head all the way along towards the tail, and then the oppositional psoas muscle just here, and that provides the locomotion through the water, that provides the up and down, that propels it through the water. And the muscle, if I just take a sample out, is extremely dark, certainly compared to terrestrial animals. So again, if I show the camera there, you can see how dark that tissue is. And Paul, I don't know if you want to explain why it is so dark.
Yeah. So what that is actually it's something called myoglobin that makes it very, very dark. Myoglobin is a pigment that stores oxygen. And so actually in the blood, for these animals, remember these are mammals, so they're air-breathing mammals. They breathe air through into their lungs. They distribute the oxygen through their lungs, through the blood, into the muscles. And then when they go on their dives, I mean, porpoises are quite shallow divers. They go to maybe 200 meters, and not much more than that, for a few minutes, no more than that. But in the species that dive really deeply, so we're thinking like sperm whales or some of the beaked whale species that can go maybe 2,000 or 3,000 kilometers for over 2 hours. I mean, the record at the moment is about 2 hours, 18 meters and nearly 3,000 kilometers on one breath of air. And that was a Cuvier's beaked whale in the Pacific, recorded just a couple of years ago. And so this is the ways that how can an animal that breathes air like we do, I mean, if we hold our breath, in a swimming pool, how long can we go? Sort of 30 seconds, a minute, 4 minutes, whatever. These animals can go a lot longer than that. So, and that is simply because they're able to have these extra oxygen stores, not just in the muscle with the myoglobin, but also in the hemoglobin in the blood. Their red blood cells are larger than ours, They have a higher hemoglobin concentration, which is another pigment that stores oxygen, and they also have a higher blood volume and a higher muscle volume. So they're really, I think the deep divers particularly, they're really using their muscles like scuba tanks. They're just charging oxygen in there when they're on the surface, and then they're really just slowly releasing this oxygen from the muscle when they go on their deep dives. And this is how they can do these incredibly long dives, particularly deep diving species.
Right.
So, and this is really only new, this relatively new aspect of science. Scientists have been working on this for many years, but these animals are so difficult to study that really a lot of these species, we only ever get to study them when they strand on the beach. So strandings have been very informative, not just for porpoises, where we see a lot, but there are species that we see very rarely that normally live a long, long way from here, usually they spend all their time at depth, maybe 2,000-3,000 kilometers feeding on deep sea squid, and those are the species that, some of those species have only ever been identified as species when they stranded on the beach. So strandings can have a very informative in about all the species that we get in different parts of the world, and actually some of these incredible adaptations like the muscle and the hemoglobin in the blood.
Well, I can see up until this point, Rob's been using what looks like a very small, but I imagine very sharp knife. But you've got another piece of equipment there now, Rob. What's you going to do with that?
Okay, that £5 B&Q. It's going to last for 25 years, so great job B&Q. So this is what we use to cut the ribs. We can take the rib cage away. That will expose the chest cavity, and we've already exposed the abdominal cavity and some of the organs that are there as well. I should have mentioned at the beginning, this has been a little bit rushed, obviously, because of the technical issues. This animal has been stored frozen, and when they are stored frozen, all the cells expand and then rupture with the formation of ice crystals, and it becomes what we call hemolyzed. So that's why the body looks quite as red as it does. When it's very fresh, as this one was before it went in the freezer, it wouldn't have been as red as this, and that's why it's a bit difficult to differentiate on the camera. So we'll next move on to taking away the ribs. This is the sound effect time, I'm afraid. Sorry, guys. So we'll cut through the ribs on the underside. Just separate those. And then around towards the cranial attachments. Okay, I'm sure that sounded quite gruesome back in the auditorium. We missed one.
There's always one at the end.
Okay. And then we can take that rib cage away. Okay, so now we've exposed all the organs. Again, Matt's documenting all this as we move on through photographic evidence in case we need to go back to it.
While you're taking
Perhaps
photos...
we can get the camera to have a look at what we're seeing internally before we move on to taking each organ system out one by one. So this is the lung. Underneath the lung is the pericardial sac containing the heart. And then this is the diaphragm, just here, running that way. Underneath that is the left lobe of the liver. And then they have 3 stomach chambers. So the current thinking is that cetaceans as a group diverged about 40 million years ago within the artiodactyl clade. Common ancestors, well, ancestral forms now might include cattle and so on. So cattle have 4 stomachs, cetaceans have 3, largely speaking. A long intestinal tract, and then we have the left kidney just here. We've also exposed the reproductive tract, so I'm pleased to say in this instance the animal is not pregnant. In my hand here I have the left ovary, which has some scars on them. Now porpoises are quite interesting because they appear to almost exclusively only ever cycle in the left ovary, hardly ever to never in the right. So I'm sure if we take out the right ovary in a second and compare them side by side, you'll see distinct differences between the two. Now here we have an enlargement of that left ovary, which is what we call a corpora luteum. And then there's the scars. These are evidence of previous cycling, production of eggs. And these are called corpora albicans. They're a rough proxy, I guess, for the aged individual. So it appears to have cycled recently, to have ovulated, although that may not have led to the production of a viable calf. And it has been pregnant in the past. We can tell that by the appearance of the uterus. We have here linear striations running along the horn of the uterus, and then a network of blood vessels here. So it has been pregnant in the past. It has cycled recently, although there's no evidence that it has been pregnant this season. Okay, so the next thing that we'll do
Rob, I might
is collect some
just stop you there
samples
for a
and-
moment, if you can hear me.
Of course.
Yeah, sorry.
Absolutely.
While you're having a closer look and Matt's finishing off with his photos, I thought this might be a good time before we go further to take any questions from the audience. So if there is anything you'd like to ask, anything we've covered so far that you'd just like clarification on, or you've suddenly started wondering about a particular aspect of Rob and Matt and Paul's work, now is the time to pop your hand up. If you can't think of anything right now, that's fine, because we'll take questions again later on. But we're also taking questions online, of course. If you send us emails to learning@zsl.org. My colleague Hannah at the front here is checking that inbox every few seconds. Hannah, do you have any questions that have come through yet? One. Let's have a little look. Okay, and those of you who are in here, if we've got one over there, brilliant. So we'll come to you next. Let's take this question that has come in from, I'm not sure if there's a name for it, but the question is, I understand porpoises are bottom feeders and are resident around the UK coastline, where they may come into contact with polluted rivers and possibly untreated sewage. How does this impact their general health and sort of contribute to disease? So perhaps, Paul, this might be a good one for you to take, because porpoises are found very close to our coastline rather than far out at sea. Are there more issues associated with sewage and things like that?
Yeah, actually, there's a lot of diseases that these animals can have. Infectious diseases, viruses, bacteria, fungal infections, as well as the parasites. A lot of the bacteria that we do see are often very specific to porpoises, that we don't really find them in humans. There have been cases in different parts of the world where pathogens, bacteria from sewage have actually ended up in marine mammals in certain areas, but I think most of the things that we find around the UK are more like, the salmonella, for example, we often find not just in the intestines of porpoises, but also sometimes in the lung. And this is, whenever we get salmonella in a porpoise, it's always just one type that's very specific to harbour porpoises. It's a porpoise salmonella that's adapted to harbour porpoises, and maybe part of the reason that we find in the lungs is that's how porpoises can, when they surface together, they can create an aerosol and they can transfer from one animal to the other. So that's how things like parasites and bacteria can survive and transmit between one animal and another. So we don't find a lot of problems with sewage as such, but I think that the sort of chemical pollutants we do find in the sediments, as we mentioned earlier, and as we talked about, the chemical pollutants can lower an animal's resistance to these parasitic diseases or these bacterial diseases, and can sometimes, in severe cases, they can actually die of these infections, particularly if they're very immunosuppressed.
Okay, well, anyone watching online, do continue to send us your questions to learning@zsl.org. But let's hear from the question in the room.
Yeah, I just wanted to ask, why do they have 3 stomachs?
Why do they have 3 stomachs? Good question.
Yeah, that's a very good
Paul?
question. Yeah, I'm not,
Do you know the answer?
I mean, that is a very good question. We don't fully know the answer to that, but partly is the evolutionary history. Rob mentioned, they evolved from animals that have multi-chambered stomachs, the artiodactyls. They're our closest relatives. And so when, they basically came from land animals into the sea, so they kind of kept these adaptations. With some species only have 2 stomachs. Some of the squid eaters have only 2 stomachs, so some of the dolphins, the deep diving dolphins like the dolphin wingings, and so a few of the larger whales only have 2. But the ones that have 3 often eat fish, and the first stomach is very, very tough. Well, Rob may show this in a moment, but what it is when the fish goes into the first stomach in a porpoise or a dolphin that eats fish, very often fish can have spines. They can have really, really sharp spines, and that's part of the fish's protection to try and stop things eating them very easily. But also these spines can actually puncture. If you've got a very soft stomach, like we do, and you eat a fish, like a horse mackerel, or for those of you who know some of the fish around in our seas, these can have really severe spines in their dorsal fins, and it's all right as the fish goes down, but then once the fish is in the stomach, and if it was a very soft, these spines could actually puncture and cause a rupture, and that can be very, very serious. So I think with the ones that eat fish, they have this very thickened first chamber, and that is partly just protective. It's the same with the esophagus. It's the same lining as the esophagus. So as it swallows the fish will go down the esophagus with this very tough lining, very keratinized epithelium, and then into the first chamber of the stomach, where then you can start the digestive process. And then after the flesh is digested down to the bones in the first chamber, then the more fluid digester can then pass through the second and the third chambers, which are soft-walled, and then through, there's other digestive enzymes that come in at that stage, and then they pass through the intestine, which is also very soft-walled. So really, it's quite sort of energetic, the demanding to produce all your intestine very thick-walled, and it wouldn't absorb very well. But if you make the esophagus in the first chamber very tough, then I think, if nothing else, it gives you very good protection against eating fish with spines. And a lot of fish don't have very tough spines, but some of them do. And we know over 25 years of doing this work that actually the list of species that porpoises or any of the dolphins eat, there's a long list. They don't just eat one type of fish. They eat a lot of fish depending on what time of year it is, where they are, and fishery friends and all sorts of things. So actually the variety is enormous of the type of individual species that they will eat. And therefore some of those will have spines, I think, and that is part of the reason why they have this very tough first stomach, which is very thick-walled and protective for them.
Well, we will see the stomach later on. And in fact, I think Rob might be sort of trying to separate some of the digestive tract at the moment. But Rob, tell us a little bit more about what you're doing now, and also, have you seen anything yet that is out of the norm, anything that stands out?
No, so while you were chatting with Paul, we were just moving on. So just to recap where we are at the moment, Matt and I have taken some standard samples for bacterial culture. So we take it from the lung, from the kidney and the liver. It's a standard set of samples we collect in any animal. We've also taken virology samples from the lung, the kidney, and we'll do the spleen in a second as well. Again, standard samples we want to collect and archive in case we want to go back to them. The next thing I'm doing is just removing each organ system one by one, looking for any evidence of abnormality. So just to pick up on what we have here, I mentioned earlier that they cycle almost exclusively, in terms of porpoises at least, in the left ovary. So actually I'll try and orient it the right way around. So pointing to this camera here, we've got the left ovary and the right, and they look very different. So we've got this corpora luteum and then corpora albicans scars, and the right is completely quiet or quiescent. So just an interesting observation for porpoises. There's a lot of asymmetry in cetaceans in general that might reflect why we see that asymmetric cycling. I've also taken out the left kidney and the left adrenal gland, and now I'm removing the organs. But to go back to your question, Charlotte, about have I seen anything unusual so far? Not really. Apart from the moderate nutritional condition, there's not anything else we've seen thus far. But if we are going to see any pathology, it's usually confined to the lungs. We get a lot of pulmonary pathology, as Paul probably said, but also in the digestive tract. So we'll see where we end up and what we find out as we go through these organ systems.
Okay, well, as you carry on, Rob, I'm sure one thing that maybe a few of you are wondering is what it smells like in there, because this is one sense that we cannot join you in here in the lecture theatre and anyone who's watching online. So I don't know if you or Matt are you able to describe what this smells like?
Like a fine wine. I don't notice it, I've got to be honest. After 18 years of doing it, you just...
It's varying smells. It kind of smells a bit like spam today, to be completely honest. Off spam. We've had stuff which will make your eyes bleed in here, to be completely honest. But yeah, it's not too bad today. It's a good one. That's the best I can describe them, I'm afraid. It just smells like a porpoise.
But if anyone's really keen to find out how they smell, meet them in the bar later on. You might smell it on us.
I was going to mention that both Matt and Rob are going to be coming back here after the event and hanging out in the bar area. And I do believe the smell can linger a little bit. So do sidle up to them later on if you're curious. Rob, what are you doing next?
So, as I tried to say earlier, pathology is, broadly speaking, the description of abnormality. We're removing each organ system one by one to look for any evidence of abnormality. So I'm taking out now the right kidney, and I'll also take out the right adrenal gland. So as well as looking for abnormalities within organs, where there are two of them, you look for abnormalities between organs as well. And I've also removed just over here, this very large organ is the liver. There are two lobes to the liver, large right lobe, smaller left lobe. And if we take some random incisions through that, we're looking for any evidence of parasites, any evidence of lesions or disease that again might indicate there's something wrong with the liver. So what I can see immediately as we open it up, hopefully on this camera here we can see there's a white fibrous tract within the liver. And if I squeeze that, this is probably the moment we're going to get lots of groans from the audience. These contain parasites, and this is a species called Campula oblonga. I'm just looking for them on the back of my glove. Little trematode flukes, similar to what sheep might have, sheep flukes. And they are quite specific to cetaceans. Okay, so I think I've got a bunch there. So on the back of my glove, you might be able to make out those little ovoid objects there. Those are the flukes we're looking at. Now, in terms of the relevance, this individual, they're probably secondary importance because there are not very many of them. There's a lot of liver that's unaffected. You can see it otherwise looks pretty, I think I've spent a word for it. It does look normal, it does. And not much liver is actually affected by these fibrous tracts. So we make a quick qualitative assessment, how much the liver is compromised. I'd say 1 to 2%, not very much. We record it as an incidental finding. And the other thing we take from liver is a sample of liver tissue that will then also, at some point, be analysed for the naturally occurring metals. So Paul, I don't know if you want to say anything about the metals we look for either.
Yes, we can look at a whole range of heavy metals, and we have done over the years. So the ones that are most toxic are things like mercury, cadmium and lead. They also can bioaccumulate, so you get higher concentrations, species higher up the food chain. We should also say, though, that there are a lot of naturally occurring metals or sources of metals in the marine environment that are naturally occurring. And for example, even the ones that the species that eat squid, like sperm whales and Risso's dolphins, for example, they often have really quite high levels of cadmium. And this is because cadmium is actually used as an oxygen transport substance in the blood of squid and cephalopods. So actually the fact that just their diet of feeding on squid gives them very high exposure to cadmium. The species that eat fish, they can get very high levels of things like mercury, and so the fish eaters, they get higher levels of mercury. And if we actually tested some of the levels of mercury in the liver of some of our porpoises that have been living around for a long time, or some of the dolphins, you could get levels of metals, of mercury in the liver that would kill everybody in this room. Probably there's so much mercury in these animals, but the thing is they're able to detoxify the mercury because they've had an evolutionary exposure to it. So over the 60 million years that the porpoises and the dolphins have been eating fish or squid, they've been exposed to these very high levels of metals naturally from natural sources, volcanic activity, hydrothermal vents, things like that. The man-made sources of these from industry only came very recently. So actually these animals have evolved ways to actually detoxify the metals. So we're probably less worried about the metals. The difference with the metals and the PCBs, of course, is the PCBs are very modern compounds. They've only really been used in the 20th century, and so these animals have not had time to evolve very good detoxification mechanisms like they have for the metals. So that is the difference. But as I say, we look at both, and actually a lot of the levels are either stable or declining, the different metals, depending on which region. But for all the chemical pollutants, probably Scotland is the cleanest in the UK. There's been less industry there, and so whether we test for metals or PCBs or other contaminants, very often the porpoises in Scotland that have the lower levels, and the ones in England and Wales that have the higher levels.
And I guess too, this highlights again why it's so important to have all of this data and over a period of time as well, because then you can start to see if there are any changes in these levels that can sort of give us an indication of what might be happening out there.
Yeah, yeah.
Okay, Rob, what happens next?
Okay, so moving on. We're going to compare the kidneys next. So I've sliced through them, and hopefully for this camera over here. And it is difficult to make out because, I say, it's quite reddened because they become frozen. If you slice them open, you can see that they have quite an interesting structure. So they have two kidneys, just like us, but again, they are very different internally. So they have what's called reniculated kidneys, or it's termed sometimes colloquially a bunch of grapes-like kidney. There are individual units within it. So hopefully the camera will pick up that there's a paler central area, a darker outer area. So it's a cortex and medulla, just like our kidneys, but they have hundreds rather than two. So it's almost like built-in redundancy, which is a much better evolutionary model. And again, there's nothing that I can see internally to indicate any abnormalities. Over here, I've got the adrenal gland, which I've cut in half. And hopefully with the camera over here, we'll be able to pick up, we have two layers. Again, we've got the outer cortex, inner medulla, and there should be a 50-50 ratio between those two, ideally, in a normal, healthy individual. But hopefully the camera will show that there's a cortical enlargement. Now, the adrenal gland is the site of stress hormone production, so adrenaline, cortisol, and porpoises seem to live a very stressful life. They're attacked by bottlenose dolphins, caught in fishing gear, exposed to marine pollutants. So as a consequence, potentially of that stressful lifestyle, they do have adrenal enlargement. There's also potentially a direct physical effect because of an association with elevated PCB levels, and that's something we found through some of our research. So adrenocortical hyperplasia, I think is a technical term, adrenal enlargement. So that's probably one of the more interesting things we've found so far. And also then taking out the cardiovascular system, which is essentially the tongue, around the larynx, both lungs and the heart, and remove that whole organ system and then remove it to a separate tray. And I think we're probably going to move over onto the smaller table in a second, because the other thing we'll do is we will probably look at the head. Because this is a live stranding, there's a chance this individual may have had an underlying infection of the brain, or meningoencephalitis would be a technical term. So we will actually try and take the brain out to show you that, and then take some samples of the brain as well in situ.
Okay, Rob, well, while you're moving things across, we might pause again to take any further questions. Anything you'd like to ask or even have a closer look at as well, if you're particularly interested in an organ in a porpoise's body, we can obviously zoom in and have a closer look for you. So is there anything anyone would like to ask in the room at this point? Hands up. We've got one on the third row here and also at the back there too. So if we go over to the back, yeah, and your microphone, because that looks closest, and then we'll come back to you in a moment.
Hi. I was just going to ask, when you initially took some samples for the population genetics side of things, it looked like you took blubber sort of skin. I just wondered if there's a particular reason for that, whereas in sort of normal human postmortems, you take a muscle sample, and forensics and things like that, you take the psoas muscle from humans.
Yeah.
Yeah,
I mean, you can use muscle. Part of the reason that probably skin is used is that often it's animals on the beach, stranded, it's very easy to take the things that are on the outside first, and just in the bottom of the skin layer, you'll get where a lot of the new cells are, and they kind of go out, probably like human skin, but the ones in the inner part of the skin have the nuclei, and then you can get the genetics from them. I mean, yes, muscle is just as good. I mean, probably better in some ways, but you can do skin or muscle. Some scientists would use muscle. One of the problems is that with people that are studying free living populations, with sort of whales, dolphins, sometimes the only samples you can get are often small biopsy darts, and that in different parts of the world people use this. It's a fairly benign process. It's a very benign process. It doesn't cause any harm to the animals, but you're just producing a little bit of skin from a little biopsy dart from a free living animal, and then you can actually tell a lot of information from that, including the genetics. So I mean, perhaps that's why when we're merging our strandings data with people who work on biopsy of free-living whales, then, you know, skin is the thing that we can all access relatively easy without actually causing any harm to the animal. Obviously, with free-living animals, it'd be very difficult to get muscle samples from them. So actually, skin is a sort of way you can get a very small sample very benignly without actually causing any suffering to the animals. So that's why we take skin samples mainly.
Good question. And then
Good
I
question,
think,
yeah.
where was the hand? There we go, got microphone, excellent.
Forgive me if I'm wrong. I've read some literature about beluga whales and the PCB and contaminants affecting them by individuals becoming hermaphrodites. I don't know if that's correct, but is that a problem in harbour porpoises or not something you see?
Yeah, that's right. Yes, beluga whales. For those who didn't hear the question, beluga whales in the St. Lawrence Seaway particularly have been a very well-studied population. There's about 500 of them in the St. Lawrence Seaway, sort of in Canada, and they've been studied for many years. And in fact, this population has been quite highly exposed to PCBs and other pollutants as well. And there's been a lot of abnormalities, things like hermaphrodite, which is kind of been sort of male and female at the same time. That's very abnormal. But it's one of the things that chemical pollutants like PCBs, because they mimic hormones, because they have this endocrine disrupting effect, you know, they are endocrine disrupting chemicals, they mimic hormones, that's part of how they have a toxic effect, that's how they can interfere with reproduction. So for example, our porpoise is not pregnant, that may be a PCB effect as well, that, you know, ideally in a pristine world where there's very low pollutants like Iceland and Greenland, where porpoises have been studied there, they literally have a calf every year. They've got very low levels of pollutants, and they're nice and fat and healthy. They have much lower levels of parasites than ours, and actually that's one of the interesting differences we see. And in these pristine environments, porpoises, pretty much if they're healthy, the adult females will become sexually mature about 3-4 years of age, and then they'll produce a calf every year. In our waters around the North Sea and around the UK and Germany and Scandinavia, you know, some of our Northern European animals, they've got higher levels of pollutants, and we only see pregnancy rates of about 50%. Only about half of our porpoises are actually, and these are the healthy ones, you know, but see, well, the ones that are healthy that die in fishing nets, the trauma cases, the bycatches, they only have about half of them are pregnant at any one time. So we still think that really the PCB is having some effect on suppressing their population because of that. But do we see hermaphrodites? No, we've never seen a hermaphrodite in a porpoise, but we do look for that, and we've been looking for 25 years, but we've not found one yet.
Any other questions before we go back to Rob? Let's take one more just over there.
Bit of a random question, but how many kilos of fish would a harbour porpoise eat?
Oh.
How
Yeah.
much?
Yeah,
how many
I
kilos?
Paul, how
yeah,
much?
I
a day?
don't know that for porpoises actually. I know I've worked it out for killer whales. For an adult male killer whale, that you can get adult males that theoretically up to about 110,000 kilograms. They're mostly much less than that, but they can eat sort of 3 or 4% of their body weight. A very active adult male killer whale. So, you know, if it was 10,000 kilograms and they eat 3 or 4% of their body weight, then that would be, you know, about sort of a few hundred kilograms of fish a day, you know, potentially.
day?
So it's enormous, the numbers for those, which is much smaller for the porpoises. I mean, we don't know. Part of the problem is that we'd love to know the answer to that, but, you know, when we have an animal like this, we do a post-mortem examination, a necropsy, we're only seeing them in a snapshot in time, so we can only see what's in the stomach now at any one time, and often if they're diseased, you don't see any food in the stomach. If they're caught in a fishing net, often you will, and that tells you what they're feeding on, but only at that time in time. But what we really want to know is, you know, on a daily basis, and as that animal is a juvenile and grows to be an adult, how much food is it eating, and how does that vary on a day basis, on a weekly basis, on a monthly basis? We'd love to know that, but we really can't. We can only really infer from what we find on these snapshots of time from a post-mortem examination. But still, I think a lot what we do know about what they eat and how much they eat has come from stranded animals, as well as actually studying them working with fishermen.
Well, we're going to go back to Rob in a moment, but before we do, we have had a few questions through from people watching online, so thank you very much. So let's see if we can briefly just give an answer to one or two of these. We've had a question from Trinity. Thank you very much, Trinity. Are there any pollutants that uniquely affect cetaceans and not other sea animals that you know of, Paul, or does it affect all of the animals out there?
I think they all can have an effect. All the pollutants that we work on all potentially can have an effect. I think the reason we're worried particularly about PCBs is because we get the highest concentrations in the cetaceans, in the whales and the dolphins and the porpoises, particularly the ones that live a long time, like killer whales that live near the coast, where they're exposed to, because these are man-made pollutants. So the ones that live near our coast and coastal waters can get the highest concentrations. So those are the ones we're most worried about because of the concentrations. But I think basically because porpoises, well, the dolphins and the killer whales are at the top of the food chain, they're often kind of quite removed from the more coastal things that you might find in mollusks and small fish in coastal areas. They were exposed to different pathogens, bacteria, sewage, things like that. So, but actually the smaller fish will be eaten by the bigger fish, then the bigger fish will eat the medium fish, and then you get the killer whales and the dolphins at the top of the food chain. And so those are the ones that, why they get, as we said earlier, the very high levels of pollutants. And of all these pollutants, they all bioaccumulate, but it's particularly the PCBs that, it's kind of like a double whammy, because the PCBs not only at higher concentration, just because there was more of them, and they're harder to degrade in the environment, but also they're the most toxic, even more toxic probably than DDT. So really it's kind of a double whammy. These animals have the highest exposures to the things that are most harmful to them, and that's why really we're really worried about PCBs more than anything else now, in Europe particularly.
Well, let's go back to Rob and Matt, because I've got a question for you, Rob, which is coming from Liam, who's watching online as well. And if you can hear me, Rob, if you just give me a little nod.
I'm here. Hello.
Excellent. So Liam's question is, how long does it take the CSIP team to complete a necropsy on larger cetaceans, like the sperm whales that stranded last year?
Okay, that's an interesting question. So in many ways, and I've got some echo on this again, so forgive me if I look a bit confused. So that's my general look, actually. So in many ways, cetacean examinations like this are more detailed than human postmortems. So if we're doing this in a fresh animal that hadn't been frozen in a lab, it could take 3 to 4 hours. Now, if we're looking at a large animal on the beach, there are a few issues. The main issues are trying to get access to them, to be honest, because you often have to get permission to examine these animals on the beach, permission to open up on the beach, and then all the logistical and other constraints that go around these large animal examinations. And actually, when it comes to the examination itself, it can be much more limited. So the blubber layer is great at keeping the whale nice and warm when it's alive. It can also keep the whale nice and warm when it's dead. So as we found with some of the sperm whales on the east coast, they have a habit of going pop or bang on the beach. Everyone must have heard of exploding whales. Some of them did explode rather impressively on us while we were opening them up. And unfortunately, what that means pragmatically is it limits what you can collect pathologically. So if you don't get to those whales pretty quickly after death, actually you can limit quite severely what you can collect at post-mortem. So that was a long, rambling answer. The short answer, Liam, is 3 to 4 hours with a porpoise in a lab. It can be even less on the beach. It really depends how fresh that animal may be.
Okay. Well, Rob, let's have a closer look now at what you've got in front of you. You've removed from the main part of the body. What do you have there?
Okay, well, I'm sort of juggling 3 or 4 bits here. So on my left, we have the cardiovascular system. So we've got the tongue, the larynx that fits into the blowhole, because obviously these animals breathe through the top of the head, and then the esophagus running down towards the stomachs. I've also opened up a couple of stomach sections as well. So if we go over here, first of all, this is the first stomach chamber. This is called the cardiac stomach. It's very thick-walled to withstand the very acidic enzymes that it produces, so pH sort of 2.5, that sort of thing. And if you open that up inside, there's actually not a lot in there. There's no really convincing evidence of recent feeding. There's some, I can feel it, beach material, some sand, some grit, and a few pebbles as well. There are actually also a few parasites. I think Paul might have mentioned this species earlier on. So on the back of my hand is a species of parasite called Anisarchus simplex. So again, to get a groan from the audience, humans can be impacted by Anisarchus as well. You can get anisarchiasis if you eat too much raw fish. And just as an aside, if you like sushi, like I do, I found this out recently on an episode of The Fat Bikers. Apparently all sushi in the UK is frozen before we consume it, because that would then kill the parasitic cysts that might live in the fish. So if you eat raw fish by the sea, then you're kind of at risk of getting these guys. Now, if we go back over here towards the esophagus, this is really what we're interested in terms of what these guys feed on. Just here in the esophagus is a small ovoid white structure. I'll lift it out and again place it on the back of my glove, hopefully, without dropping it. So again, for the camera, the white object is what we call an otolith. It's the ear bone of the fish. And they're quite species specific. So we can take that and send it to one of our colleagues who examine those and work out what they've been feeding on whilst they're alive. And this brings it back to the point I was trying to make earlier, beyond trying to establish the cause of death, which we'll hopefully get towards knowing a bit more about later on. We're also interested in how they've lived. So collecting material like this will tell us more about what they've been feeding on and so on.
Fantastic. So what other areas of the organs can you look at next, Rob, to see if there's anything out of the ordinary?
Okay, so we'll go back to the lungs now. We will remove the esophagus, which there's a few otoliths in there. Okay, we'll cut that away. Just as an aside, this is the aorta. That's the major blood vessel that comes out from the heart. So that's the one that David Attenborough says you can swim up in the blue whale. As an aside, historically we have had blue whale strandings in the UK. The Natural History Museum have been collecting data on strandings in the UK since 1913. And that's a really interesting and valuable resource because we can see changes in distribution of strandings over time, and actually changes of some species that no longer strand in the UK. So we have had a handful of blue whale stranding in the UK, all in Scotland, none since the mid-20s, and that's largely because of the major commercial pressure of whaling, which has had significant impact on whale populations out there. The good news, and there is some good news to be drawn from studies like these, obviously, is that some whale populations are beginning to recover. We banned most commercial whaling, most of it, in 1986, and actually it signed into effect in my hometown in Brighton in 86. And now, over the last 30 years, some of those populations of whales are beginning to recover. And the one we always use as an example of that is the humpback whale. Because this historical time series in the UK going back 100 years, we had no humpbacks at all stranded in the UK over the first 80 years. Since that ban on commercial whaling, we've begun to see strandings, and we've had about 25 or so, I think, in the last 25 years. So it makes a broad point that actually strandings can be good news. They can tell us, and that's quite paradoxical, they can tell us that there are more of those species out there. So that's certainly the case for humpback whales. Some species that we don't see stranding, though, that we might expect to see them, and I think Paul will talk about killer whales in particular, if he hasn't already. Those are species where we have much more cause for conservation concern.
Yeah, Paul was telling
so
us
while I've been
earlier
chatting,
on, Rob, about the strandings and how they can sometimes be a good news story rather than
exactly
a bad news story.
Okay, cool.
What are you seeing inside the lungs?
So as I was repeating what we said earlier, I was just opening the airways up. So we're going to compare again left and right lungs. And again, if we open those up, lift them up towards the camera, we can see again parasites in the airways. So we know from previous work on porpoises they have 3 species of parasite that we find in the airways. These large ones here, which I'll just lift up for the camera, and that's breaking away. These are called Pseudalius inflexus. And then there are smaller species of parasite called Toroniurus convolutus. And then in the lung tissue itself, there'd be a third species, very, very small, that lives in cysts called Halocircus invaginatus. So there are 3 different species of parasite we find in the airways. And what we'll do is make a very qualitative assessment about their relative burden. So I would say, with the benefit, hopefully, of 18 years' experience, that's probably a moderate burden of nematode parasites in the airways.
Yep.
Matt will take a picture again.
Yeah, Paul's Paul's
And we see
nodding his
them-
head in agreement with you, Rob.
It's always gratifying. So we see them ubiquitously in porpoises. You're just trying to compare relative burdens. Now, if I squeeze the lung tissue itself, it feels quite rubbery. There are some nodular areas, and the head of the parasite would actually attach in the lung tissue itself and cause some localized reaction. But actually, most of the lung tissue feels not too bad. And again, it is quite red because it has been frozen. But beyond those moderate parasite burdens in the airways, the lung tissue itself feels okay. And then we'll flip the lungs over, and we'll then look at the blood vessels, the pulmonary arteries, to look at the relative parasite burdens within those. And that will take me a few seconds to locate, so talk amongst yourselves.
So again, if it is all getting a little bit too bloody for you, do feel free to close your eyes or just step outside for a moment. But while Rob is cutting away there, are there any more questions from inside the room, as it were? Anything else anyone would like to
Red on me.
ask? We've got a hand up, I think, at the back there. So Linda's just coming to you with a microphone. So let's take a question there.
Hi, have you seen much evidence of the microbeads in these animals that's been in the press recently?
Yeah. So just in case anyone didn't quite catch that, that was asking about any evidence of microbeads, because there's been quite a lot in the press about them being banned in face scrubs and things that you buy in the shops, because these microbeads, very small plastics, can obviously then get into the environment.
I'll deal. I'll pick that up. Because I'm going to come on to the stomachs next, and we can talk about that as well. So, just going back to the lungs for a second, there are some parasites in the blood vessels too, and we'll dissect those further down in a second, perhaps a bit later when we have more time. But it naturally leads on to the stomach. So we've been contracted for the last 12 years to look routinely for evidence of macroplastic in the stomach. So in this individual, we can't see any. And actually, it's certainly the case with the stranded cetaceans we've looked at for the last 25 years. We've had 3,500 post-mortems. Only one animal where the cause of death has been due to macroplastic ingestion. And certainly, I can't recall any case where we've seen the beads you're referring to as well. So at least on the basis of the UK Strandings programme, plastic ingestion and plastic impacts does not appear to be a significant issue. One of the things that you might have heard about in the audience and at home as well, of course, is microplastics. That's where fragments of plastic break up in the water column, become ingested potentially, and they could be an issue, because the idea is that the microplastics, the small fragments of plastic, actually act as absorbing agents to increase the ingestion of other marine pollutants like PCBs. Now, we're only at the beginning of that process of trying to look at that story. We have a collaboration in place with colleagues at Exeter University. So we'll collect the whole gastrointestinal tract, freeze that will go off to them. They'll actually sieve through the contents, look for any evidence of microplastics that you might find in the gastrointestinal tract, and then from that, we'll try and do some further research to work out whether microplastics are having an impact. But just to recap again, macroplastic ingestion is not a significant issue, at least from what we've seen in UK stranded cetaceans.
Okay, so what other evidence or information are you looking for in the stomach and kind of digestive system there, Rob? Okay, so we talked about the cardiac stomach. This is where digestion takes place. So the fish would go in sort of head first, because interestingly, when they from the fish, they throw them around, so on the head first, so the spines don't get stuck in the esophagus. So the fish go in here, and then the second stomach, this is the glandular part. You can see it looks very different. If you open up the stomach wall, is quite folded, but it's a very different appearance. This produces the enzymes that then get refluxed back into the first stomach. So the flesh is stripped off from the fish fairly quickly, and then the soup, if you like, then goes back through into the third stomach. You add in bile, it goes to the intestinal tract. Now, for those who were there for the first event we held last year, you might recall, we opened up the cardiac stomach, there's a mass of bones in that individual, and that was probably the causal factor in the death of that animal. Not the case here. So just some beach material, a few ocelots, a few parasites, not very much going on. In the second stomach, and this might not be very obvious to the camera, I can feel some small lumps and bumps within that stomach wall. Again, this is indication of another species of parasite. This time it's called Campula oblonga. I may be able to show you if I cut through those, but it appears to be relatively heavy. And as I'm cutting through it, I can feel some
calcified, cutting against the knife, and that tells you it's a chronic, long-standing condition. So there's some calcification and organization around those Phileta parasites. So it's got a moderate to heavy burden of parasites in the stomach wall. Again, to some extent, we're trying to work out the significance of that finding. We do find them ubiquitously in a lot of porpoises. It can cause an issue when it can become so extreme it would actually cause an obstruction in the communication to the next stomach, and that's what I'll go on to next, is trying to work out whether that's caused an obstruction anywhere. Doesn't appear to have done because my finger will actually go into the next stomach. So again, it's probably an incidental finding.
Okay, Rob, well, while you're continuing to sort of sift through there, we have had another question from Victoria, who's watching online. She wondered how you can estimate the age of porpoises, and in particular this porpoise, if you have an idea as to how old it is, and also how long harbour porpoises tend to live on average.
So I can, again, to the magic of television, I can feel that, if that's okay.
Yeah.
On the back of my hand, if the camera shows that, I've removed a section of teeth already. So again, this is another set of samples we collect. We collect 2 sections of teeth, and that's how we age these animals. They have annular growth rings in the tooth, just like you have growth rings in a tree trunk. And that will be sent off to another set of colleagues, another set of collaborators. The tooth will be fixed, cut in half, and then stained, and you'll actually count those lines to work out how old that individual is. And again, because our audio and video cut out earlier, I'm not sure if you covered this already, but porpoises probably live to around 20 years old. So one of the shortest lived species of cetacean on the planet. And again, just as an interesting aside, people might not know this, the oldest species that we know about at the moment is the bowhead whale, which is an Arctic species. A lot of Arctic species are quite slow and therefore long-lived. And a couple of individuals there have been aged some interesting methods recently. One individual aged 212 years old, so they can be immensely long-lived in some instances. But the porpoise, quite a short-lived species in contrast.
Fantastic. Thank you very much, Rob. So we won't know for sure how old this animal is until you get that information back from the teeth.
No, and a lot of what we do is in collaboration, to be honest. So here we're carrying out the gross pathology, probably gross in more than one sense of the word, and then that together with some data from next door where the bacterial cultures will be carried out over the next few days. That will hopefully give us a broader picture of how the animal died. But a lot of what we do and collect goes into our archive in case somebody might want it in the future, because you can't go back and collect it again if you want it after the fact. So we do collect things opportunistically in case you want to go back and analyse them in the future. A lot of these analyses are carried out in collaboration, so it can take many months, sometimes actually years, to get the data back. But it will be produced over time, hopefully.
Rob, is there anything else you're going to look for in the digestive tract at this point? Any more evidence?
Yeah, so I'm going to go on to the third stomach in a section, that's the pyloric stomach, and I've also begun to open up the intestinal tract at several random locations. So we're just seeing here, it's a quick look-see to see what there may be within the intestinal tract. Also, if there's any evidence of tapeworms, perhaps, that might indicate, be evidence of perhaps why this animal died, but I haven't seen anything so far. And I also haven't seen any evidence of recent feeding. You will see some beige-coloured fluid, which is perhaps digester, evidence of recent feeding. So this individual has nothing in the stomachs, nothing in the intestinal tract, perhaps some bile staining. So again, the camera might not pick this up. If I scrape the contents and rub it on the back of my glove, they're actually stained green, which is evidence of bile staining. They continuously produce bile, which helps fat break down in the diets. And so that continuous bile staining is evidence of a period of inanition or lack of recent feeding. So again, it's evidence this animal has not been feeding for some time. Period of time I don't know, to be honest, it's speculation to say more about it. But why it hasn't been feeding, we haven't actually got to the bottom of just yet.
Okay, well, Rob, I know you're keen, before the event finishes, to move on to the head so that we can have a closer look at that. But before we do, and while you continue to have a look for anything else obvious, does anyone here in the audience have any final questions for Rob and Paul? Anything you're still wondering, they will be in the bar later, so you can grab them then too. But we've got a hand up on the back row, so let's take a question there, and we'll maybe take one more from online as well.
I was just wondering, because of the historic data set with the strandings, is there any influence in terms of more males or females washing up? Because maybe that would affect the population growth in the future.
That's a very good question. And actually, interestingly, we see an identical 50-50 split, at least certainly in harbour porpoises where we've got the most data, 50-50 between males and females. So that's what you would predict, and we're not seeing any biases in that. And I think some of the dolphins as well, we see pretty much a 50-50. The common dolphin is our next most commonly stranded species, the common dolphin, and we get a lot of bycatches in that species as well, adults and juveniles, and we tend to see a 50-50 split as well. So there doesn't seem to be any biases, at least in terms of the age, the males and females that you would predict. Of course, you can have biases with stranding data, so we should say that it's not a perfect system, and very often we can find very interesting things about the species, their causes of death, their health, their pollutant exposure, and how these things change over time. But actually, we also need to know what's happening at the population. So, for example, with bycatch, in the early 90s, we found and identified dolphins and porpoises getting caught in fishing nets as a major cause of death. But actually, we can't just predict what the population level impact in the free-living population will be just from working backwards from our stranded animals, because there are biases in the data. So that's why DEFRA and the government also fund other scientists like the Sea Mammal Research Unit in St Andrews to actually work with the fishermen to quantify the bycatch in harbour porpoises and common dolphins and other species at the population level. Alongside that work, where we're working with these problems, and where we think we have a bycatch problem in a particular species, the government also has been funding research to try and mitigate that, whether that is putting devices on fishing nets like pingers that can actually deter animals from being caught, or other gear modifications to try and reduce the bycatches as well. So actually what happens is, over a 25-year period, we identify bycatch as a cause of death initially, then subsequently we find it every year in our study as a large cause of death. So not just in porpoises, but in common dolphins and other species, and this happens in other parts of the world. So then actually researchers, also ecologists and other scientists, work with fishermen to actually try and work out what the population level impact is, which fish, and different types of fishing gear catch different types of species. So the ones that get set on the bottom, the gill nets, they tend to catch harbour porpoises, whereas the midwater trawls tend to catch things like common dolphins. So different species going for different types of fishing, going for different fish, cause also different types of...
Sorry to interrupt, but we're just going to bring up one of the slides, because as Rob makes his final preparations there, a question we have had in from Esteban, who's watching from Chile, kind of links to that, because he wondered what were the most common causes of strandings in harbour porpoises. And this chart sort of nicely summarises a lot of your findings over the past sort of 24, 25 years. So what are the main things that we're seeing?
Okay, well, you can see, if you can all see this, and hello to Esteban, it's great to have you with us all the way from South America. That's absolutely fantastic. So this is for harbour porpoises, so this is just the different causes of death over the period of the project, and we can actually see that number one is these different infectious diseases. So these are the parasites, like we see, the bacterial infections and other things like that. That is that category. There's porpoises, we do see a lot of infectious disease compared to some of the dolphins where we see less, but and this may be a part of where PCBs is also having an immunosuppressant in maybe some of these cases, not all necessarily, but some of them. The second on the list is interspecific interaction. This is actually porpoises being attacked and killed by bottlenose dolphins. If you look at the second picture here on the right at the top, you can see that's actually a bottlenose dolphin attacking a harbour porpoise, and actually in areas where we get the two species together, in Scotland in the Moray Firth and in West Wales, you actually do see these porpoises, and I think we can actually show a video of a porpoise actually in Scotland, one of the very first cases documented where The large bottlenose dolphins are actually attacking, and you can see in the slow-mo here, the little porpoise, almost like a rag doll, just being tossed. And actually now this is one of our most common traumatic causes of death in harbour porpoises. But we only really see it in 3 regions, in Scotland on the east coast, in West Wales, and around Cornwall, where there's a few bottlenose dolphins as well in that population. So that's really interesting. The third one down is bycatch. We've talked about bycatch. This is accidental entanglement in fishing gear. The fishermen don't want to catch harbour porpoises or dolphins, but they occasionally do, as we've talked about, and a lot of research is being funded in the UK by the UK government and in other countries to try and mitigate this problem. And I think it is being mitigated, actually. It is actually getting better, which is why now I think bycatch was always the most common cause of death for many years, and actually it is going down now. So I think this is part of the funding that the government puts in for projects, not just ours, but a lot of other people who do work on trying to use technology to limit the bycatch is actually getting an effect.
Really
and
good
so
news.
it's a good news story. It's not all bad, that where we are finding things that are very important, that actually science and technology and research funding can actually help the problem. And this is a good evidence actually that now the porpoises killed by bottlenose dolphins is actually, we've seen more of them now than we have of the bycatchers. It never used to be the case.
Well, we're going to go back to Rob, who is now hopefully fully prepared with all his protective gear and the like. And Rob, yeah, you're looking good. Why is it important
Safety first.
to look at the head? What information might this give you?
Well, I think I said earlier that because it's a known live stranding, there's certainly a possibility that has an underlying brain infection. I mean, if it has, that would certainly explain why it hadn't been feeding for some time. So we are going to try to remove the brain. That being said, there's certainly some indications that it may be on the, how should we call this technically, on the runny side. So I'm not convinced it's going to be successful. So if that's the case, I'm going to actually then move on to showing you very quickly how they echolocate, just as an added bonus. So if you're ready, here we go. So I'm going to use the circular saw just to cut around over the top of the skull and then over the back as well. Okay. Got the screwdriver, Matt?
Yes, over there.
Great, thanks. Everyone still with me?
I think
Good.
so far, Rob.
Okay, now it is on the runny side, so I'm not actually going to try and take it out because it'll probably fall apart in my hands, but hopefully I can show you here, and then we'll collect a sample in a second. We'll take a swab of the brain and some samples for virological analysis as well. We have 2 halves of the cerebrum. That's the conscious control, the ego, the self, the id, whatever you want to call it. Underneath that is the cerebellum with the spinal cord just protruding there. That's the unconscious control, so breathing, perhaps echolocation to some extent, all those things controlled there. So we'll take a sample of the brain tissue as well. It certainly doesn't look abnormal. There's no evidence of trauma or traumatic injury, so certainly some kind of traumatic strike doesn't appear to be a likely factor here either. So Matt will just take a quick sample.
Rob, we are coming towards the end of the event. So from the information you've gathered so far, has that been able to tell you, sort of give any suggestion as to why this animal might have stranded, or are you very much going to have to wait for the laboratory results to come back?
It's in the latter camp, yeah. I mean, that is often the case with these examinations. So let's recap where we are, and then if we have time, I can show you how they echolocate as well. We have an adult female harbour porpoise that we know live stranded and died on the beach last August, been stored frozen since then. Moderate nutritional condition, no evidence of recent feeding, so nothing in the gastrointestinal tract to tell us it's been feeding recently. Some moderate parasite burdens in the airways, in the lungs, and in the liver too, and some light burdens of parasites in the stomach. So there's nothing really here that, to me at least, is a smoking gun, if you like. So I think we're certainly going to be dependent on follow-up analyses, some bacteriology, cultures, and so on, to try and perhaps flesh out the story of why this animal may have live stranded and died. But again, just it's important to make the point that this is the individual unit of the project. This is the N=1. That's what we build the rest of the research on. The long-term nature of this project has been funded by UK government for 25 years. It means we have a huge amount of data that will hopefully allow us to address these questions of conservation concern and try and do something about it in the long term.
Okay, Rob. Well, we are coming towards the end of the event. I'm sure people watching online wouldn't mind hanging on for a couple of more minutes to see how these animals echolocate. But just by a kind of nod of hands, is everyone okay just to sort of sit still for a couple more minutes just to see this little bit? Yeah, you're okay with that. All right. Rob, do you need to prepare the head any further to show us how they echolocate?
I am good to go.
You're good to go. Okay,
Let's
then,
do it.
Rob.
Yeah.
Tell us.
Because I think everyone's desperate for a beer. I know I am.
Tell us how it works.
Right. Okay, so let's go back over here first of all. So we go back to the lungs and the airways first of all. This is the larynx. The larynx fits into the blowhole. Around the larynx is a muscular sphincter. That's the initiation of echolocation. So cetaceans dive on inspiration. They go, and then dive down because they need the air to actually produce the sound they make. So the muscular sphincter contracts and relaxes, and it sends short pulses of air up into the blowhole. So going back to the skull, blowhole just here. I'll put my finger up there, and I'm not going to ask you to do it, the audience, but I can feel a bifurcation here, so that splits it into two channels. So there are two kinds of cetacean. There are those called odontocetes, those with teeth, and then the mystocetes, the baleen whales. All odontocetes have this soft, fleshy structure called a melon at the front of the head. The brain, as we can see now, is here. So actually, the pulse of air then goes past a set of structures called the phonic, or monkey lips. And if we can get the camera in tight, hopefully over here, I can show what I mean by that. These soft, fleshy pads just there, if that's hopefully obvious. I'll try and move it around again. Those are the phonic lips. And so what happens here is they're held under elastic tension against the front of the skull. As the pulse of air goes past, it pushes them away, it displaces them, and because they're held in place with ligaments under quite tight elastic tension, they snap back. And without trying to get blood on my face, as they snap back, you get a sound. And you send pulses of air. Suddenly you've got a click train that we all know Flipper makes. So that's how they start to make the noise. Now in this species at least, they then have air-filled baffles just here. These are called vestibular nasal sacs, and there's one here, one over there. They insulate that area, prevent most of the sound coming up. Most of it is then pushed forward through the melon. And I've taken this section away. If we take that away, the melon is a very specialized area of fatty tissue, again with muscular attachments, the red bits underneath. The muscles will contract and relax, and it changes the shape of that ball of fat. So essentially it acts like a focusing lens. It's very highly vascularized, highly specialized bit of tissue for cetacean. So it modulates the sound wave going through the melon, changes its pitch and frequency and so on. So it goes out towards the target fish and then comes back again. So essentially using echolocation in much the same way that Second World War submarines were using. So you have the ping, boo, going out and the return. And then the distance, time it takes to go back, tells you how far away that prey species might be. And the current thinking is actually the sound is channeled along the lower jaw, along what we now call acoustic fats, towards the ear bones or tympanic bulla, and the sound is then received there. So it's a really highly specialized, highly discriminate form of biosonar, way, way in advance of anything that man's come up with so far in terms of technical achievements that allows it to echolocate, to navigate in the water. And that's the main sensory adaptation that cetaceans have, is this form of echolocation.
Fantastic.
Is that all right?
Yeah, thank you so much, Rob. Well, I'm afraid that does bring us to the end of this evening's event. Thank you very much for all of your questions, though. For those of you who are here, Rob and Matt are going to be running down the road and making it to the bar as soon as possible, and Paul will also be joining them round the back. So please do feel free to hang around. The bar's open until just before 9, and have a further chat with them, ask them any more questions that you might have. For those of you who have been watching us online, thank you so much for joining us, and thank you for your questions as well. If we weren't able to answer your question during the event, we will try and send a reply to you by email, so you still get an answer. And certainly, if we bring up the PowerPoint just for a moment, if you would like to find out about sort of the results of this case, as it were, do keep an eye on the CSIP Facebook page, because we'll pop kind of the results up there, sort of update you probably via that page, but also we'll try and send you an email, because I think if Jen's around, I think she might have all of your emails here in the room. So we'll send you an email as well to tell you what the results are of those laboratory findings, and whether maybe that gives us an answer to why this animal stranded. Those of you in the room, I believe you should all hopefully be holding onto an evaluation form. Please, please let us know what you thought of tonight's event, because it's only your feedback that can help us hopefully program more events like this in the future, and also sort of change them for the better. And if you're watching online, you too can give us your feedback as well. You should be able to see a link to our feedback forms on our webpage, so please fill that in as well. So thank you once again, everyone, for joining me. But if we bring Matt and Rob up once more, we can give them a big round of applause, and also Paul as well. Thank you all so much.
Right?
Thank you very much, Paul.